Hello guys. Nephrons are known as the fundamental structures of unit. They are known as the structural and functional unit of the kidneys which are involved in the filtration of the blood leading to the formation of urine. Today we are going to discuss what is a nephron, how nephrons are organized, what are the different components of nephron and how mammalian nephrons differ from that of the avian or the bird's nephron. Nephrons, as I told, they vary in number from animals to animals, but usually they range between 1 to 2 million per each kidney in human, and around 40 millions of nephron are seen per each kidney in case of the cattle. So it depends on the animals and it also varies. In our next lecture, I will tell you the diversity of nephron among different domesticated animals. But we are going to focus on an average, that is the nephrons of the human or common in mammals. As I told, nephrons, they are the structural and functional unit of the kidney. So when I am saying it is the structural component, that means they are forming the major bulk of each kidneys. And when I'm saying they are known as a functional unit, that means they are associated with the functions of the kidneys. Let's make it little bit more detailed. The structural component, it shows various parts like the glomerulus and the Bowman's capsule, which are said to be the fundamental part or the first part of the nephron, which is known as glomerulus and Bowman's capsule. They are commonly known as the Malpighian body. Malpighian body, or you can say this as Malpighian corpuscle. These are the part which are involved in the process of, and that makes the functional part. These are associated with the process of filtration of blood, and due to this filtration of the blood we are getting the ultimate product that is known as urine, and this urine is the source of removal of non-gaseous nitrogenous waste and metabolites from the body. The second component under the structural part is the renal tubules. We can also use the word nephric tubules. Renal tubules or nephric tubules. These renal tubules are further divided into proximal convoluted tubule, the longest part of the nephron, not the loop of Henle, that is PCT is the longest part. The second part is the most important part, as you know, that is loop of Henle, commonly known as LOH, having three distinctive parts: descending loop of Henle, ascending loop of Henle, and hair clip part of the loop of Henle. We'll discuss. Then the distal part, that is known as distal convoluted tubule. The proximal convoluted tubule is the major site of absorption. Remember, around 70% or more the nephric filtrate are absorbed along the PCT. One of the most important part of water absorption and salt retention and concentration of urine is loop of Henle. And then the distal convoluted tubule comes, which absorbs water and electrolyte, but in a facultative manner, because they require the presence of the hormones like ADH or vasopressin and aldosterone for the secretion for the reabsorption of water and the salts. After the DCT, then the lower part comes, that is collecting tubules. Collecting tubules further by the collecting ducts. The collecting tubules are the structures where the secretion occurs. That means those substances which are not required in the urine, they will come back from the lumen through this collecting tubule to the interstitial fluid, and those which are required to go in the urine, they will come from the interstitial space or the cellular space into the lumen. As a control mechanism, collecting ducts are responsible for acidification of urine. We will discuss in detail. So these four parts, they are coming under the word renal tubule or the nephric tubule, and which are associated with two major functions, that is selective reabsorption, and the third process, that is known as tubular secretion or the augmentation. So here this word filtration of the blood is otherwise known as the glomerular filtration. Glomerular filtration always occurs under a pressure, and that pressure is known as the effective filtration pressure, and we'll discuss in our coming lecture how this pressure is generated. The second part is the selective reabsorption. As the name indicates, it is selective in nature. That means not all the substances are going to be reabsorbed or retained. Rather, substances preferentially they are going to be reabsorbed on the basis of their concentration, permeability, and as well as few molecules against their concentration gradient. That's by the active mechanism or the active transport mechanism. And once the selective reabsorption is over, then the tubules secrete few of the components into the urine so that the final formation of the urine will be made, and eventually the urine will come out through the collecting tubules ducts to the ureter to the renal pelvis, and where the urine will be stored. So this classification shows that the nephron is having two distinct parts, that is structural component, these are the structures, and of course when we are saying about the functional part, we are discussing these three important functions, and they come under the common word that is known as urine formation. So with this, we finished the introduction to the nephron as a structural and functional part. Before going further into the details, please like the channel and subscribe the channel for this type of content and easy understanding of physiology. Let us discuss what are those nephrons which are characteristically present in the mammalian kidneys, and how these nephrons are varying from the nephrons which are present in birds. These are important questions people will ask you for better understanding and answering the questions in entrance examination. So let's see. So typically in case of mammals, two types of nephrons are present in the kidneys, and these two nephrons are structurally different and also they are functionally different. In the course of evolution, two carry out the process of water conservation mechanism, or with the respect of water intake and output, these nephrons have been evolved, and these nephrons are important and they function depending on the need of the kidneys. But we should not forget, because there is a myth is there, that these two types of nephrons they form different types of urine. That's wrong, because in both this type of nephron, the collecting ducts and collecting tubules collect the same, that's important, same nephric filtrate, and ultimately it drains into the renal pelvis. So that's why do not mistaken it as that these two type of nephrons are fundamentally different at the terminal level. So let's discuss vividly what are those type of nephrons. So the mammalian nephrons, basing on their distribution, structure and function, they are classified into two types. One is known as the typical mammalian nephron sometimes, and depending on the position, this is known as cortical nephron. In few animals, they may be absent, just like the dog and cat, where it is almost 0%. And the second type of nephrons are known as the juxtamedullary nephrons. Juxta means junction. So just at the junction of the cortex and the medulla, some of the nephrons are present, and they are known as juxtamedullary nephrons. Almost they are 100% in the kidneys of the dog and the cat. So it clearly says that depending on the animal species also they vary, but within the species they are almost in the constant zone. So now to see the fundamental difference between these two type of nephrons. So as I am discussing today about a common human kidney, or you can say a common mammalian kidney, in general we are not specifying anything. So that's why this is around 80 to 85% of the total nephrons, and this is 15 to 20% of the total nephrons. Exceptions already I told. These cortical nephrons are mostly confined to the outer part of the kidney, which is histologically known as the cortex, outer cortex and inner cortex. And juxtamedullary nephrons, as the name indicates, it is present at the junction of the cortex and medulla and more towards the medulla. These type of nephrons, the glomerulus, the Bowman's capsule, and PCT, DCT, they are being present mostly at the cortex. Students, try to understand, in a kidney, this part is the outer part which is said to be the cortex, and this is the medullary part, and this is the renal pelvis where the ureter arises to carry the urine. So when I am saying the glomerulus, the Bowman's capsule, PCT, DCT, they are present here in the outer cortex, whereas the loop of Henle, which is around 18 size, it hangs down from the cortex to outer medulla. That means the medulla, which is not towards the renal pelvis, rather towards the cortex. And this loop of Henle only remains here up to this region, which is known as the outer medulla. If I can draw a diagram up to this is known as outer medulla, where osmotic gradient is not that much. But when we are coming to the inner medulla, then the osmotic gradient increases. Now coming to this kidney, that is juxtamedullary nephrons, here the Bowman's capsule, glomerulus, DCT, PCT, they are present no doubt at the corticomedullary junction, no doubt, but the most important is that the loop of Henle, and that is the determining factor. This loop of Henle goes more in depth, that is, it passes from the outer medulla to inner medulla, and that is more towards the renal pelvis. It means the concentration gradient and the osmotic gradient increases. The osmotic gradient increases. That means these type of nephrons are more in concentrating function. They also increases the osmotic index of the filtrate because they are present in the deep seated. We will discuss this during the time of countercurrent mechanism and countercurrent multiplier system. But in this class, just I wanted to convey you that these two nephrons are fundamentally different. Their location and their presence and their distribution inside the kidney make them to function differently. Now coming to these cortical nephrons, they are usually responsible for, no doubt, for urine formation, and that too concentrated urine formation. That's not a problem. But the thing is that these juxtamedullary, they form more osmotically active urine. We will discuss what do you mean by that, by osmotically active, that in the next lectures. So more osmotically active urine, and that's why they sometimes are said to be the concentrating nephrons of the kidney. But we should not forget the DCT, PCT, the loop of Henle, all are similar in case of both of these. Little bit, the juxtamedullary loop of Henle is longer and thinner in the membrane. That makes it more concentrating in function. But the collecting tubules and the collecting ducts, particularly, they are similar to both these type of nephrons. That's why they do not do any further change when the nephric filtrate reaches the collecting tubules and collecting ducts. So this is all about the mammalian nephrons. As I told, we will see a diversity class in my next lecture. There I will tell how different animals show different type of nephrons and what's their number. But the take home message, that is the fun fact number 1, that juxtamedullary nephrons are almost 100% in case of dog and cat. But in general, they are 85-15% in distribution in other mammals. Now, I am comparing these nephrons with another species. So you can say in the evolutionary course, this has been developed. As you know that most of the mammals, terrestrial mammals, they are ureotelic in nature. That means they excrete urea in their urine as the non-gaseous nitrogenous waste. But birds are so specialized that they want to conserve the water as they take very less amount of water and they do not have the sweating mechanism. So therefore, to maintain the salt load and water balance, they have evolved special type of nephron in them, which makes them very specialized. But we should not forget that their nephron structures are also almost similar to that of the mammals, but in the course of evolution, two types of nephrons have been developed. So now let's see the difference between the mammalian and the avian nephrons. So this flowchart shows the avian nephron, the nephrons which are found in the birds. So here we are seeing the two types of nephrons are there, and this makes it very important, that is reptilian type of nephron, which is not found in case of mammals, and mammalian nephron. That means just like that of the mammals, these nephrons are. This reptilian type of nephron are so specialized that their loop of Henle is absent. So this makes this nephron very specialized. Loop of Henle is absent. The proximal convoluted tubule is connected to the distal connecting tubule by means of a remnant or a evolutionary old type of collecting tubule, or simple I can say tubule. And here this makes this very specialized, that is reptilian type of nephron. As well, mammalian type of nephron means they show the same structure just that of the mammals. That means the PCT, the DCT, the important loop of Henle, the collecting tubule, the collecting duct, and of course the Malpighian corpuscle or the Malpighian body. And here also the same Malpighian corpuscle or the body is present, but here the loop of Henle is absent. Can you believe? It is so interesting that in case of birds, it has been seen depending on the salt load. Sometimes reptilian type of nephrons, they become functional, and sometimes the mammalian type of nephrons become functional. They are interchangeable in their function, and it is only to conserve water, conserve electrolytes in their body. So the take home message, that is the fun fact number 2, that is loop of Henle is absent in the reptilian type of kidney, reptilian type of nephron, which are the parts of the avian nephron type. With this particular lecture come to the conclusion. My assignment for all of you is that please write down 5 comparative points between the cortical nephrons and the juxtamedullary nephrons, and number 2, 5 comparative points between the avian nephron, that is reptilian type of nephron, and mammalian nephron, and the third, compare the mammalian nephron with that of the avian nephron. So in the next lecture, I will tell you the answer with these comparative points. So thank you so much and keep learning. Please subscribe the channel and like the content for the growth of the channel. And we are coming with the part 2 of this lecture with 3 comparisons which we are going to discuss. So stay tuned for the next lecture. Thank you so much and have a wonderful time.